Today's lecture is on cell death and cell renewal. So we're going to discuss then in this chapter programmed cell death or apoptosis, a little bit about stem cells, pluripotent cells, etc. So cell death and proliferation are balanced throughout our lives, and our cells are turning over at a pretty rapid rate. Animal development involves a lot of cell proliferation and differentiation, but also cell death. When your fingers and toes are initially going through development, there's webbing in between, and cell death, programmed cell death, actually leads to getting rid of the webbing in between. So it's very regulated. In adults, it balances our proliferation and keeps our cell numbers constant. Also, it's important to get rid of cells that could potentially be dangerous to us, such as virus-infected cells. So this is all in red, so things that you guys want to start to study. Necrosis, that would be when you have accidental cell death from acute injury, and sometimes toxins can cause things like that as well. Apoptosis is programmed cell death. And there's three things I'd like you guys to be aware of that will happen or can happen during apoptosis. One is DNA fragmentation. The second is chromatin condensation. And the third is fragmentation of the nucleus and the cell. So if you take a look at this here, guys, you can see in the DNA fragmentation and chromatin condensation, there's a big difference that you're going to see in the nucleus. Eventually, the nucleus starts to fragment, and eventually you get this blebbing that occurs down here, and you get what are called apoptotic bodies. And this is a great example of that, guys. Here's your normal nucleus here. You can see that they're all broken up. The cells are starting to fall apart there. Also, this is a great example, guys, of what happens to the DNA. So up here would be high molecular weight DNA, and you can see as time goes on, you're getting fragmentation that's occurring. So basically, these fragments then are phagocytized by our macrophages, and they're removed from tissues. Now, with necrotic cells, they will swell, and then they lyse, and then their contents are released into the extracellular space. This quite often then is going to cause inflammation. So the apoptotic cells express eat me signals such as phosphatidylserine. Normal cells, the phosphatidylserine is really restricted into the inner leaflet of the plasma membrane. So as that membrane starts to break down then, guys, it signals the cells to kill themselves. So here's your normal cell, here's your phosphatidylserine. As the cell starts to undergo apoptosis, however, that serves as a signal to bind to other receptors, and then apoptosis is going to take place. Now, a lot of the studies originally done on apoptosis were done in C. elegans, and C. elegans is a worm, and its development includes death of about 131 specific cells. So it's easy to study. They reproduce relatively quickly, which is why they were used. So at the time that they were studying this, and this was a while ago, they started noticing these different what they called SED proteins that led to apoptosis. SED3 then is what we call a prototype of Caspases, and this is now what we refer to these proteins as, Caspases. They're a family of proteases. So again, any protease, guys, is going to break down a protein. So Caspases have a cysteine residue at their active sites, and they will cleave after aspartic acid residues in their substrates. So they're pretty specific for what they do. So they're pretty much the ultimate executioner, guys. They cleave over 100 different target proteins, and there's an activation of what we call initiator Caspases that start kind of a chain reaction, if you will, of activation. So again, these are all the different targets that we have here and kind of what they do. DNA, laminin, cytoskeleton, Golgi, and scramblase, I like the name of that, which basically affects translocation of the phosphatidylserine in the cell surface. So they're synthesized as inactive precursors. So you'll see in a lot of the literature, pro-caspase, whatever, that will actually be converted to the active form when you have cleavage then of the pro site. So in C. elegans again, there was a protein that was studied called SED9, and in mammals that's actually BCL2. And what that does, I'd like you guys to know, is it inhibits apoptosis. So what happens then is cancer cells then will be unable to undergo apoptosis. So any cells that express this cannot undergo apoptosis. So there's about 20 proteins related to the BCL2. Some actually inhibit, and others will actually induce caspase activation as a result. So this is kind of the family here of BCL2. We have two that are anti-apoptotic. We have two that are pro-apoptotic effectors, Bax and Bak. And these are only pro-apoptotic here. That group right there. So please know that Bax and Bak are downstream effectors that directly induce apoptosis. So when I say downstream, guys, if you look at the membrane, and then what happens as you go into the cell, anything that goes downward, points downward, is downstream. So these are all inhibited by anti-apoptotic BCL2. Now the ones that we showed on the last slide that say BH3 only, they're upstream. So they're higher up in this cascade then. And when activated by death signals, they antagonize then these BCL2 and activate Bax and Bad. So if you inhibit or you have an anti-apoptotic situation, actually apoptosis then is going to occur. So again, here's a good example. So here's your death signal. When I say downstream, guys, I'm talking about anything down here. You get activation of the BH3, and then that activates then all these other proteins that will lead then to apoptosis. Now in mammals, the BCL2 proteins act at the mitochondria. So the mitochondria really plays a central role. Now what Bax and Bak do is they induce then cytochrome C. If you guys remember when we talked about electron transport, there were cytochromes that were involved in that. The cytochrome C then will trigger caspase activation. Now caspase 9 is a very important type of caspase. They're all important, but this one is important because it forms a complex with what's known as APAF1 and cytochrome C. And that whole complex then of these two is called an apoptosome. So here's your death signal. Bax and Bak get activated here, guys. They will go into the mitochondria. Now again, you're going to get cytochrome C that gets released, and this will form a complex then with caspase 9. So this wheel-looking thing is the caspase 9. And then when the little blue balls, which are cytochrome C, come on, it's the apoptosome. And that's a better view of that. So then another type of caspase called caspase 3 gets involved then. And it's a pro-caspase at first. Then it gets cleaved, becomes active. And then that leads to apoptosis. So they're also regulated by inhibitors of apoptosis, and there's a few of those that are out there. I like the name of this one, Reaper and Grim. Okay, so here's your death signal. Reaper and Grim then are going to be inhibitory. They inhibit the initiator and effector caspases, and then you're going to get the apoptosis. Okay, so there's a lot of different types of programmed cell death, and it's mediated by different signaling pathways, and we've talked about signaling pathways in the last couple of chapters. Intrinsic pathways are activated by DNA damage. Extrinsic are activated by signals from other cells. So a key player in programmed cell death, guys, is p53. And it will actually arrest cells in response to DNA damage. So that's what its effect is. We also have a couple of other players in here, ATM and another protein kinase Chk2, and they will phosphorylate and stabilize p53, resulting in increases in those levels. So again, when you've got p53 there, you're going to arrest the cells, induce apoptosis so that the damaged cells will not continue to divide. So here's your ATM, and you go through Chk, that's phosphorylated. It's a kinase, phosphorylates p53. You get more p53. Ultimately, apoptosis is going to ensue. Now, please know that the major signaling pathway that promotes cell survival is initiated by PI3 kinase. And I talked about this in another chapter that will activate a threonine kinase called AKT. And then downstream of that, there's a whole cascade of other phosphorylations that will take place. Now, we have two other types of phosphorylations that can take place. Phosphorylation of Bad and FOXO. I actually have a paper published where I looked at bad phosphorylation. And these will maintain them in an inactive state. So again, guys, just to give you the big picture here, here's your growth factors. PI3 kinase gets activated. That goes on to phosphorylate then AKT. And there's a couple other players here. Then Bad gets inhibited, FOXO gets inhibited, p53 over here will get regulated as well, and our good friend NF-kappa B, and you can inhibit translation on that side. So it's a whole big picture. There's a big balance in our cells between survival and cell death. So our extrinsic pathway, guys, tumor necrosis factor we talked about before, so I'm not going to go into it that much more, but NF-kappa B can be regulated by that. And the receptors activate another initiator caspase called caspase 8. So again, tumor necrosis factor stimulates. Here's caspase 8. Eventually, the Bax and Bak get into the mitochondria, and then you get your complex over here.